May I have your attention, please? Our network is down again, so our webcast is not yet working. So if anybody's listening through some other mechanism, I hope IPTV might be recording. IPTV folks, if you can be recording so we can broadcast or webcast later, that would be helpful. We're going to go ahead and start timely anyway, and we'll just resume when we can. So sorry about that, and I hope we get the webcast going shortly.
Good afternoon, and again, we are having a little bit of difficulties with our webcast, but we're going to go ahead and get started with the presentation. So, good afternoon, good evening, or good morning, depending on from when and where you are joining us. I'm Dr. Phoebe Thorpe, and it's my pleasure to welcome you here to the CDC Public Health Grand Rounds for March 2017, Emerging Tick-Borne Diseases. We have a very exciting session, so let's get started, but first, a few housekeeping slides. We do offer continuing education credits for public health grand rounds for physicians and pharmacists, nurses, veterinarians, health educators, and others. Please see our website for additional information. This is the disclosure slide for this session. We're also available on all your favorite social media websites. Please send your comments and questions to the Grand Rounds email box at grandrounds@cdc.gov. We also have a featured video segment called Beyond the Data. This month's segment features my interview with Dr. Bobby Pritt. It'll be posted about a week after the session. We have also partnered with the CDC Public Health Library to feature scientific articles related to tick-borne diseases. The full listing is available at cdc.gov/scienceclips. Here is a preview of our upcoming Public Health Grand Round sessions. Please join us live or on the web at your convenience. In addition to our outstanding featured speakers, I'd like to take a moment to acknowledge the important contributions of the individuals listed here. Thank you. And now for a few words from CDC's Acting Director, Dr. Schuckett.
Well, thanks so much. It's actually liberating knowing that we're not webcasting yet. Millions of Americans seek care for a tick bite each year in the United States, and despite that, very few of us are equipped to answer the questions from friends and relatives that are in that category. Today, you will be hearing that the reported cases of tick-borne diseases are increasing, that the range of ticks that can carry diseases is expanding, that the number of tick-borne diseases that we are aware of is increasing, and that the laboratory approaches to figure out what they are is also increasing. If that list is making you feel full, wait until you see the pictures of the ticks who have been feeding during this lecture. I believe it is the most memorable part of the slides you'll be seeing. We're in for a treat because I think with the four speakers and the slides that they've prepared, most of us will be better equipped to handle that tick that you might find on you yourself or a loved one and to help the nation be more prepared. Thanks.
Thank you, Dr. Schuckett. And now for our first speaker, Dr. Eisenson.
Thank you for the introduction, Phoebe, and good afternoon. I'm going to start with some background information. All known tick-borne infectious diseases are zoonoses. Ticks can maintain the pathogens through transmission to their offspring or acquire infection through feeding on an infectious host. Importantly, humans are incidental hosts that are infected through the bite of infected ticks, but they do not serve as a significant source for infecting other ticks or perpetuating the pathogen's life cycle. Ticks are unique among arthropods in the diversity of pathogens they transmit. Among the 18 tick-borne disease agents described to date in the United States, 14 are bacterial and are transmitted by 5 tick genera. However, at least 3 unique tick-borne viruses transmitted by 3 tick genera and at least 1 tick-borne protozoa have been shown to cause human disease. In the following slides, I'll present data to show that the majority of vector-borne diseases in the U.S. are tick-borne, that in recent decades, the number of tick-borne disease cases has increased. The geographic range over which tick-borne disease cases have been reported has expanded, and a growing number of tick-borne disease agents have been recognized to cause human disease. Among the nearly 50,000 cases of locally acquired, nationally notifiable vector-borne diseases of humans reported annually to the CDC from states and the District of Columbia, approximately 95% are transmitted by ticks. Notably, Lyme disease accounts for the majority of reported vector-borne disease cases, with over 30,000 cases reported annually. Without denying the significance of Lyme disease, the focus of this grand rounds will be on other human pathogens transmitted by ticks. Of the more than 84 tick species described in the United States, roughly a dozen are frequent human biters that are capable of transmitting human pathogens. Human-biting ticks are present across the contiguous United States. However, for simplicity, here I'll show the generalized distribution of only 3 human-biting ticks that are responsible for the majority of reported tick-borne disease cases. Ixodes scapularis, the black-legged tick, is primarily a woodland-associated tick that's distributed across most of the eastern United States, and it serves as a vector of the agents that cause anaplasmosis, babesiosis, Borrelia miyamotoi disease, ehrlichiosis, Lyme disease, and Powassan encephalitis. Notably, an individual Ixodes scapularis can carry multiple disease agents, thus stressing the importance of co-infections for diagnostics and prevention of Ixodes scapularis-borne diseases. The Lone Star tick, Amblyomma americanum, has a similar distribution to Ixodes scapularis, but has a more southerly distribution. It's also primarily woodland-associated and serves as a vector of the agents that cause ehrlichiosis, tularemia, and heartland virus disease. The American dog tick, Dermacentor variabilis, is among the most broadly distributed human-biting ticks in the U.S. It's found primarily in grasslands. Its range spans across the eastern U.S. and along the Pacific coast, primarily in California. It's a vector of the agents that cause Rocky Mountain spotted fever and tularemia. When looking at maps of nationally notifiable tick-borne diseases shown by location of residence, there are obvious regional differences that are explained in part by tick distributions. Notably, although Ixodes scapularis is present across the eastern U.S., reportable Ixodes scapularis-borne diseases such as Lyme disease, anaplasmosis, and babesiosis cluster in the Northeast, the Mid-Atlantic, and the upper Midwest, where humans encounter the tick more frequently than in other parts of the tick's range. In recent decades, the numbers of many notifiable tick-borne diseases have steadily increased. For example, the average number of reported Lyme disease cases has roughly tripled from 1992 to 2015. Likewise, the number of reported cases of anaplasmosis, ehrlichiosis, and spotted fever group Rickettsiosis have steadily increased from 2000 to 2015. In addition to increasing case counts, the geographic distribution of several tick-borne diseases is expanding. To illustrate this point, the map on the left shows the distribution of reported Lyme disease cases in 2001 compared with 2015, shown on the right. Although less prevalent, the geographic distributions of other Ixodes scapularis-borne diseases mirror the distribution of Lyme disease cases. Tick-borne organisms are increasingly being recognized as human disease agents. This timeline shows when tick-borne pathogens were recognized as causes of human disease. Over the first 60 years of the last century, 7 tick-borne pathogens were recognized to cause human disease. Common colors on the timeline represent a common tick genus that serves as a vector. In more recent decades, the rate of pathogen discovery has accelerated, with 11 additional pathogens described to cause human disease since 1960. Notably, more than half of those were discovered after 2000, and the majority were associated with black-legged ticks, Ixodes scapularis. There are several explanations for why tick-borne diseases and tick-borne disease agents are increasingly being recognized and why case counts are increasing in number and are being reported across broader geographic extents. Improved diagnostics and clinical recognition of tick-borne disease agents and tick-borne diseases will be discussed in subsequent presentations. Here I'm going to discuss the importance of expanding geographic distributions of vectors and a lack of effective prevention strategies. The accelerated discovery of Ixodes scapularis-borne pathogens may be attributable to an increased focus on this vector after Lyme disease was discovered in the early 1980s. In addition, though, the geographic range of this tick has expanded considerably over the last 2 decades. From 1996 to 2015, the number of counties in which Ixodes scapularis is considered to be established has more than doubled. The expanding range of Ixodes scapularis also helps to explain the expanding distribution of counties classified as high incidence for Lyme disease. Over the last 2 decades, both have expanded following similar spatial patterns. Another contributing factor to the increase in reported numbers of tick-borne diseases is increasing human contact with ticks that comes with landscape changes. There are no human vaccines currently on the market in the United States to prevent tick-borne diseases. Although we have several options for prevention, we lack a single effective, widely accepted method for preventing tick-borne diseases. Current prevention strategies fall into 3 broad areas: personal protection, environmental modification, or tick suppression. Personal protection strategies focus on avoiding tick habitat and use of repellents, specifically 20 to 30% DEET on exposed skin, and wearing permethrin-treated clothing. Daily tick checks and removal aim to remove the ticks before pathogen transmission occurs. To accomplish this, CDC recommends bathing or showering as soon as possible after coming indoors. Checking yourself, your children, your pets, and your outdoor gear for ticks, and removing them promptly. And tumble drying dry clothing on high heat to kill any ticks that remain on the clothing. Other strategies aim to modify the environment to make it less suitable for ticks. Human contacts with ticks may be reduced by using strategic landscaping techniques. Tick abundance is reduced through the use of acaricides and biological agents. Other strategies aim to reduce the abundance of important hosts for ticks in areas where humans are likely to encounter the ticks, or to reduce the number of ticks on hosts through the use of acaricide applied to the host. Finally, developing technologies focus on delivering vaccines or antibiotics to rodents to reduce the infection rates in the hosts and ultimately in the ticks. Each of the prevention strategies I've described vary in efficacy and in acceptability. Despite the large number of intervention strategies, we lack a single effective, widely accepted method for preventing tick-borne diseases. Looking to the future, it's likely that advances in molecular pathogen detection and bioinformatics, coupled with a sustained interest in tick-borne diseases, will lead to the discovery of more tick-borne diseases and tick-borne disease agents. Reforestation, increasing abundance of deer, which are important hosts for many tick species, and changing climatic conditions have led to the expansion of several vector species. Models suggest that some tick species ranges are likely to continue to expand over time, and this may lead to tick-borne disease cases being reported from new locations. A single tick species, or even an individual tick, can carry multiple disease agents. Therefore, co-infections will continue to be important for diagnosing and preventing tick-borne illnesses. Finally, there's a need to develop effective approaches to preventing tick-borne diseases. But perhaps an even bigger challenge is how to deliver effective prevention strategies to large numbers of people and ultimately reduce the trend of increasing tick-borne diseases. It's now my pleasure to introduce Dr. Paddock.
Thanks, Becky. In the next few minutes, I'll be touching on a few themes mentioned previously in Dr. Eisen's talk, using 2 rickettsial diseases as examples. The beautiful place in the left panel is the Bitterroot Valley, located in the far western part of Montana. It's not particularly large, only about 120 miles long and 10 miles across at its widest point. But at the turn of the 20th century, a deadly and previously undescribed disease emerged in this remote and sparsely populated valley. It was associated with a very high fever and a petechial rash that covered most of the body. And it killed the majority of persons who became infected, generally within the first 8 to 10 days. Of the 343 cases identified in western Montana between 1880 and 1909, 62% ended in death. This case fatality rate rivals or exceeds some of the worst infectious diseases. Investigators soon learned that the disease, which was given the name Rocky Mountain Spotted Fever, was transmitted by ticks and became the first recognized tick-borne disease of humans in the United States. The severity of Rocky Mountain Spotted Fever is linked to the tropism of the causative agent Rickettsia rickettsii to cells known as endothelium, which line the small blood vessels of every major organ and tissue of the body. This is represented by the histologic image on the left, which shows rickettsia within the endothelial cells of a patient who died from Rocky Mountain Spotted Fever. This disease progresses rapidly to involve blood vessels in the skin, as shown by the petechial rash in the center, as well as those in all of the major organs of the body. The damage to blood vessels caused by these bacteria results in extreme vascular permeability, which can lead to death when it involves the lungs and the brain. The panel on the right is a histologic section from the lung of a patient with fatal Rocky Mountain Spotted Fever that shows diffuse pulmonary edema caused by leaky alveolar capillaries. Fortunately, tetracycline class antibiotics can cure the disease if administered in a timely manner. Discovered in the mid-1940s, tetracycline drugs considerably reduced the case fatality rates to contemporary estimates of approximately 5 to 10%. Doxycycline is considered the drug of choice for all tick-borne rickettsiosis, but to achieve the most favorable outcome, it should be administered early in the course of the illness to prevent irreversible organ damage. Today, the majority of cases of Rocky Mountain Spotted Fever are reported from a belt of states in the central and southeastern U.S. that extend from Oklahoma eastward to North Carolina. The principal vector of Rocky Mountain Spotted Fever in this region is Dermacentor variabilis, also known as the American dog tick. But I also want to draw your attention to a region of eastern Arizona, which reports some of the highest incidence rates in the country and where there are no Dermacentor ticks. Beginning in 2003, epidemic levels of Rocky Mountain Spotted Fever were recorded from several American Indian communities in eastern Arizona, where the incidence rates approached 150 times the national average. CDC investigators, working closely with tribal partners and the Indian Health Service, soon determined that the brown dog tick, Rickettsiosis sanguineus, was responsible for these outbreaks. Enormous populations of brown dog ticks had proliferated among free-roaming dogs in these communities. Because dogs serve as an amplifying host for Rickettsia rickettsii, and because Rickettsiosis sanguineus is an efficient vector, this created a perfect storm for peridomestic transmission. This also represented a paradigm shift, not only in the magnitude of case numbers, but also the involvement of a tick vector not previously considered relevant in the epidemiology of this disease in the United States. Through a collaborative endeavor involving multiple groups, a community-based intervention was undertaken in a highly impacted community. The yards of 550 homes were treated with an acaricide spray, and tick collars impregnated with a long-lasting acaricide were placed on over 1,000 dogs. These efforts resulted in a marked reduction in on- and off-host tick numbers, and most importantly, a 43% reduction in the number of cases of Rocky Mountain Spotted Fever. So, for more than 100 years, Rocky Mountain Spotted Fever was considered the only tick-borne rickettsiosis in the United States. However, scientists were aware of other tick-borne rickettsia, like the one reported in 1939 from the Gulf Coast tick, also known as Amblyomma maculatum. 65 years later, this rickettsia, known as Rickettsia parkeri, was isolated from an ill patient in Virginia. During the subsequent 10 years, approximately 35 additional cases of Rickettsia parkeri rickettsiosis were identified across 9 states in the southeastern U.S. At first glance, it's not difficult to see how some patients with Rickettsia parkeri rickettsiosis might be mistaken as cases of Rocky Mountain spotted fever. The distinction between the rashes, particularly during the early stages of the disease, can be subtle. Both are maculopapular, but the rash caused by Rickettsia parkeri is generally more sparse and often associated with small vesicles or pustules, as seen in the lower right-hand panel. Nonetheless, almost all patients with Rickettsia parkeri rickettsiosis have a distinctive lesion known as an eschar, which is a scabbed necrotic area about 1 centimeter across, which represents the site where an infective tick inoculated Rickettsia parkeri into the skin. For reasons that are still unclear, an inoculation eschar occurs only very rarely with Rocky Mountain spotted fever. So, as you can see, these diseases share several clinical features, such as fever, headache, and rash. Where they differ is in the frequency of an eschar, but most importantly, in severity and outcome. To reiterate, Rocky Mountain spotted fever is a life-threatening illness with contemporary case fatality rates about 8%, whereas Rickettsia parkeri rickettsiosis is a far milder disease with no known deaths. So how much spotted fever in the United States is caused by the respective agents? We currently don't have a good answer to that question, but there is some evidence to suggest that there is more Rickettsia parkeri rickettsiosis than we know. For example, we know of multiple instances of individual clinicians who are adept at recognizing this disease and have identified up to 5 cases in just a few years of searching. Another clue comes from the relative rates of infection in the tick vectors with each of these pathogens. Rickettsia rickettsii is rarely found in dermacenter ticks in the eastern United States, and current estimates suggest that fewer than 1 in 2,000 ticks actually harbor this pathogen. In contrast, Rickettsia parkeri is commonly found in Amblyomma maculatum, with 1 in about every 2 to 4 adult Gulf Coast ticks infected with this agent. Finishing up, the take-home points from this segment are the etiologic spectrum of rickettsiosis in the U.S. has expanded during the last 15 years. Rocky Mountain spotted fever and Rickettsia parkeri rickettsiosis share many clinical features but differ considerably in severity. And finally, and perhaps most importantly, doxycycline is the drug of choice for all tick-borne rickettsiosis and in all patients of all ages. Therapy needs to be initiated immediately based on a presumptive diagnosis. Thank you, and at this point, I'm going to turn over the podium to Dr. Greg Ebel.
Thank you. Today, I want to provide you with some information on tick-borne viruses. Most of us think of tick-borne disease as fairly badly neglected in the current environment, and tick-borne viruses are neglected even among these neglected pathogens. This lack of attention is somewhat strange, so I want to take a minute to give you a sense of the wide diversity of viral agents vectored by ticks. These agents belong to a wide array of taxonomic groupings. They also have very different strategies for storing genetic information and packaging that information into infectious particles. Like other arboviruses, outcomes of human infection with tick-borne viruses are variable, but may be quite severe and often fatal. The other notable point that I'd like to make here is that tick-borne viral diseases are found all over the world, so tick-borne viruses really are a global problem. So why is it that ticks are such great vectors of viruses? If we think about the characteristics that impact the reproductive rate of an arthropod-borne pathogen, ticks have them all. They can be extremely abundant in certain areas and tend to focus their feeding on a very restricted set of host animals, often on a single host species. They also are extremely long-lived, especially in comparison to mosquitoes, with life cycles of most taking several years to complete. That means that they tend not to die before a virus infects their salivary glands and is released into their salivary secretions. There are also some other biological factors that have to do with the way that their saliva suppresses host immunity in order to permit the prolonged feeding process ticks require, and with the way that they digest their blood meals, that are all critical in making them outstanding arbovirus vectors. So in the next few slides, I'm going to review a few emerging and really interesting tick-borne viral diseases that are of relevance to us here in the US. And I'm going to start with the one that in my mind is the most problematic and about which we know the most and has the greatest potential for emergence, and that's Powassan virus, also referred to as deer tick virus in portions of the literature. Powassan virus is maintained in nature in at least 3 fairly distinct transmission cycles. The first has been known since the 1950s and involves woodchucks and a species of tick, Ixodes cooki, that's generally confined to woodchuck burrows and feeds almost exclusively on woodchucks. A second cycle involves squirrels and a different species of tick, Ixodes marxi. This cycle was also noted shortly after Powassan virus was discovered. More recently, it's become clear that a distinct genotype of Powassan virus can be maintained in a deer tick white-footed mouse cycle alongside the agents of Lyme disease, human babesiosis, and human anaplasmosis. The extent to which viruses spill over from one transmission cycle to another aren't really well understood at this point, but based on virus genetic studies, it appears that at least the deer tick-associated cycle is isolated from the others. Also notable is that most, if not all, recent human cases of Powassan virus infection appear to be linked to the deer tick-driven transmission cycle. That is part of the reason that Powassan virus is considered an emerging health threat in the US, and there is in fact some evidence to support this. The first piece of evidence is from wildlife studies. This shows that deer collected in Connecticut from 1978 through 2010 had an increasing likelihood of carrying antibodies directed against Powassan virus. The likely explanation here is that more ticks in the environment equals more tick bites to the deer, which equals more transmission, which also equals more risk, because generally human risk is proportional to the intensity of enzootic transmission, which in this case is clearly increasing. There's also good clinical evidence that the incidence rate of Powassan virus in human beings is increasing, although this could partly be due to enhanced recognition and detection. Given what's known about increasing enzootic transmission that I showed you about in the previous slide, it seems prudent to consider the possibility that human infections are indeed becoming more common. This table, showing clinical features of a case series collected from 2013 to 2015, demonstrates this. Also, please note that the case fatality rate of approximately 25% and the prevalence of severe long-term sequelae among survivors, which was about 33%, is consistent with what we thought we knew about the severity of the infection dating back to the earliest studies of Powassan virus in humans. Since 2006, 68 cases of Powassan encephalitis have been recognized and reported. Because this disease can initially look like other illnesses, it's likely that only the most severe hospitalized cases or deaths are the ones that are being reported. Cases have occurred in the Northeast and Upper Midwest, which, of course, is consistent with transmission by deer ticks. Severe Powassan virus disease occurs due to neurological involvement. These images show that this can occur via 2 distinct mechanisms. The first is due to inflammatory changes within the perivascular and parenchymal portions of the brain, which are shown in panel A. The other mechanism by which Powassan virus causes disease is direct neuronal injury, which is shown in panel E. In this particular case, nearly all of the Purkinje cells, which among other things control motor functions, were targeted by Powassan virus and were nearly absent on this biopsy. So both direct infection of neurons and inappropriate inflammation due to infection contribute to pathogenesis in human cases of Powassan virus. So we keep learning about new tick-borne viruses, so I want to take a moment to discuss 2 that are cause for some concern. I'd like to stress that very little is known about these viruses. The first is Heartland virus, a fleabovirus that was first recognized in 2009 due to 2 cases that occurred in Missouri. The virus is transmitted by the Lone Star tick, Amblyomma americanum, which is currently expanding in its distribution. Wildlife studies suggest widespread distribution of the virus in the southern and southeastern United States. Another newly discovered virus is bourbon virus, a Thagotovirus that is known from a single case report in 2015. Based on its phylogenetic position and in vitro replication studies, it seems highly likely that this also is a tick-borne virus. The sole case resulted in a fatal outcome. Again, though, the real point is to highlight that we're always finding new pathogenic tick-borne viruses and that there's a wide taxonomic array of agents that can potentially emerge. So these are things that we really need to maintain vigilance about. I want to close by taking a moment to talk about the state of the field. I mentioned that this is a neglected area, but it's not because we don't have interesting, relevant things to work on. It's been clear that much of the emergence of tick-borne diseases in our country is linked to the reforestation of the eastern U.S., which has led to the broad ecological changes we've seen in this region. So where are the knowledge gaps and unmet needs that we should be trying to fill? Well, first, there's some lack of clarity in the field regarding what's required to facilitate virus perpetuation in nature, and by extension, what factors impact when and how these new tick-borne viruses emerge. What's clear is that many different transmission modalities exist, and that in some cases, many may be required to allow the viruses to survive. A general problem in the field is the disconnect that exists between theoretical, experimental, and field-oriented individuals, and this is currently hindering progress. A second opportunity exists as we've experienced an explosion in knowledge about tick functional genomics that's arisen due to fundamental changes in the way that we collect and analyze genetic information from arthropod vectors and their pathogens. The work that's been done recently builds on years' worth of work that's characterized the potent salivary secretions of ticks, work that's continued to raise several important questions on how tick saliva impacts pathogen transmission. Those who work on functional genomics will appreciate the large amounts of data that can now be generated incredibly rapidly. I, however, would stress analysis here because it turns out that tick genomes are incredibly large, almost as big as the human genome, and that much of the information they contain is duplicated in some way or another. The functions of many tick genes are not well understood, as you can see from this figure from a paper describing the transcriptome of the tick Hyaloma marginatum. 45% of the transcripts in this case are classified as unknown. It's also, of course, become clear that many important phenotypes are controlled not only by the host genome, but by the assemblage of other organisms that inhabit ticks and all of us. Clearly, piecing this all together is complicated, but new advances in computational and experimental biology are already allowing us to make some progress in this area. So we're at a very exciting time in the history of the field of tick-borne virus disease research. We have a broad expertise in the ecology of ticks and their hosts, improvements in vitro and in vivo systems to study tick-borne viruses, and new sequencing and computational tools to apply to these important problems, but progress isn't being made as fast as we would like. There are both technical and environmental reasons for this. Many tick-borne viruses, as I mentioned, are highly pathogenic and require BSL-3 or even BSL-4 containment for study. Here's a picture of my friend Dennis trying to work on tick-borne viruses at BSL-4, and as you can imagine from the picture, that might present a few problems. Also, the systems that we're talking about are complex, as they are for all arthropod-borne disease, but the understanding of ticks lags far behind that of mosquitoes. This is partly because of the prolonged life cycle of ticks. They live longer than the grants many of us hope to obtain to use to study them. So the point is that our system in the US for funding biomedical research in academia isn't particularly well suited to work on ticks and the pathogens they carry. Finally, while tick-borne viruses are clearly emerging, they tend to do so much more slowly than do mosquito-borne arboviral diseases, which emerge in these explosive epidemics that we've seen repeatedly in recent years. So there aren't many people left working on them. So thank you for your attention. I hope that I've convinced you that tick-borne viruses are emerging health concerns, that we have interesting, relevant, and tractable questions, and we have great opportunities to move the field forward, but that we still do have some difficult technical and environmental barriers that are impeding our progress. So at this point, I'd like to turn the podium over to Dr. Bobby Pritt from the Mayo Clinic.
Thank you, Greg, for that introduction. So far in this program, we've discussed the clinical and epidemiologic features for some select tick-borne diseases. I'm now going to shift gears to talk about advances in laboratory detection methods for diagnosing tick-borne diseases. So let's first start with an overview of the primary methods for diagnosis of tick-borne diseases that we have available to us. And I'd like to actually start by emphasizing the clinical evaluation of the patient that occurs by members of the healthcare team. This is essential at this point that tick-borne diseases be considered in the differential diagnosis. And this is not just so that the correct laboratory test can be ordered, but also that empirical antimicrobial therapy can be begun if indicated. And as we heard from Dr. Paddock, this is especially important if rickettsiosis is expected, or ehrlichiosis, or anaplasmosis, because some of these infections can be rapidly fatal and they need to be treated quickly, often before the test results are available. So now let's move on to specifically laboratory methods, and these can be further divided into indirect and direct laboratory methods. Indirect methods, as I'm covering on this slide, don't detect the organism itself, but rather the host's immune response to infection. And typically this involves detection of IgM or IgG class antibodies in serum or other specimen types. Now, serology is the method of choice for diagnosing many tick-borne diseases. That's especially true for infection with Rickettsia, Rickettsia, and Anaplasma species, and there are commercially available options for these tests. Now, tick-borne virus serology is a little bit more challenging because there aren't any commercially available tests. Instead, these methods use laboratory-developed tests, and these are available primarily through the state public health laboratories or the CDC. I also want to mention that serology is not the primary diagnostic method for babesiosis. Instead, blood smear should be used. Now, one very important caveat about serology is that the sensitivity varies by the time that the specimen is obtained during the course of illness. And I want to illustrate that here on this chart. So this graph shows the general patterns of IgM and IgG antibodies following infection. And if we consider day 1 to be the onset of infection, then you can see that IgM antibodies start to rise in the first few days. They're usually detectable by 7 days, and then titers eventually drop off after several months. Then production of IgG develops after IgM, and levels will continue to rise and then can remain detectable for years. So as you can see with this graph, there's this first week lag where serology is an insensitive method for detecting tick-borne diseases, but by the second or third week, sensitivity increases significantly. So now in comparison to indirect methods, let's talk about direct methods. These detect the organism itself or some component of the organism. So for example, microscopy can be used to detect bacterial clusters of Anaplasma phagocytophilum, relapsing fever Borrelia spirochetes in peripheral blood or CSF, or Babesia parasites within red blood cells. Also, nucleic acid amplification tests are a method for directly detecting the organism's DNA or RNA. And then culture is also a direct method. It's not routinely used today except for detection of Francisella tularensis, the causative agent of tularemia, but it is an important research tool. So let's go back to our graph now to look at the utility of direct laboratory methods. In comparison to antibodies, which the curves here show, DNA or RNA can almost always be detected earlier on in the patient. This is just a generalization. You can see different curves with different organisms. Now, the onset of symptoms is also quite important when you're considering molecular testing, since this will influence when patients present with treatment or for treatment by their physician. So for many tick-borne viruses, as I've highlighted here in this yellow-orange color, you can see the onset of symptoms does not significantly overlap with the period of time that the patient has detectable DNA or RNA in their blood. So by the time the patient presents to a healthcare provider for evaluation, nucleic acid may no longer be detectable in the body. So in general, this explains why nucleic acid amplification methods can be one of our earliest detection methods for some organisms, yet can also be an insensitive method for other organisms. They do happen to be our most sensitive tool for detecting Anaplasma phagocytophilum, Rickettsia species, and Babesia species, particularly in whole blood, because the DNA of these organisms is present usually in high amounts during the stage that the patients are symptomatic. Now, there are a variety of nucleic acid amplification methods available. Unfortunately, none are currently FDA cleared or approved for in vitro diagnostic use at this time. Now, let's discuss specifically one aspect of nucleic acid amplification methods. That's called real-time polymerase chain reaction, or commonly known as PCR. By using one of several different types of probes, DNA is detected as it is amplified. And when designed well, PCR allows for sensitive and specific detection. The greater amount of nucleic acid that's present in the specimen, the faster it's detected, and therefore real-time PCR also provides some measure for how much DNA is present in the specimen. We can get even more information out of the test reaction by incorporating a step called melting temperature analysis. This can use either nonspecific DNA binding dyes or specific probes. This occurs after amplification, and melting curve analysis can alert the user if there are any mutations present. This could be a simple mutation, or it could represent an entirely new organism. And I want to show you an example of that on this next slide. In my laboratory at Mayo Clinic, we have a real-time PCR test that targets GRO-EL. That's the gene that encodes the heat shock operon of Rickettsia species. Now, we designed the primers to amplify a region of the gene that's conserved in all our Rickettsia species, and then we differentiate the two human pathogens, Rickettsia ewingii and Rickettsia chaffiensis, by melting temperature. And you can see the melting peaks here. This is possible because we chose probes that target the regions of DNA where there are 2 base pair differences. So you get 2 distinct discriminating peaks for each of the organisms. Now, what this was really interesting for this assay, as we were using it for several years, we then noticed this peak right here in the middle. Now, this peak indicated a new organism that was clearly outside of the range of Ehrlichia ewingii and Chafiensis. This prompted us to perform some additional testing, and we recognize this as a new organism now called Ehrlichia mirus oclarensis. So now let's just take a step and talk a little bit more beyond single-plex PCR, because this is talking about just a single PCR reaction. Some of the things that take us beyond that, one of them would be multiplex molecular panels. This is when you use a combination of primers and probes to detect multiple bacterial, viral, and parasitic pathogens in a single test. We already have these for respiratory pathogens, gastrointestinal pathogens, and now there are tick-borne pathogens that are also panels that are under development. One aspect that takes us a little bit beyond that even further would be broad-range sequencing. Unlike a multiplex panel where you only detect what you're looking for, broad-range sequencing, you're targeting a specific gene, you amplify it, and then there's subsequent sequence identification. We have common targets such as 16S, the rRNA gene for bacteria, and the internal transcribed spacer region for fungi. Unfortunately, at this point, there's no equivalent for viruses, so instead we tend to target groups of closely related viruses, such as flaviviruses. Now, the last category of emerging technologies that I find particularly exciting is metagenomics. And with this method, amplification of all nucleic acids that may be present in a specimen are amplified. That could be bacterial, fungal, viral, parasitic, and human. Because of the large amount of nucleic acid that's amplified, you then need to do extensive pre- and post-processing steps to target the areas of interest and remove non-relevant nucleic acid. So this is currently very expensive and time-consuming, but I think this is bound to change in the future. So with that, I'd like to close with this last slide showing the recently published guidelines for diagnosis and management of tick-borne rickettsial diseases, and thank you for your attention.
Thank you, Bobby. We're going to open it up for questions now. In the interest of time, I'd just like to ask you to keep your questions succinct and focused on the scope of the presentations from this grand rounds. I'm going to start by asking if there are any questions from online or remote sites.
We actually do have questions that came in, and again, our apologies for future. This is recorded and posted on the website by Friday. This is a technical difficulty we haven't experienced before, but we did receive a couple of questions. One that I think is a good one. How do you remove a tick?
Carefully. Bobby,
Sure, I can answer that. Well, the best way to remove it is using fine-tipped tweezers or forceps if you have them, and you just grasp the tick as close as possible to the skin and then just pull it out slowly in a smooth, continuous motion, trying not to twist it or squish it, because then that might possibly inject some of the contents of the tick into the skin. And then you can dispose of it and wash that bite site afterward.
okay, any questions from the audience? We'll take next, but let's...
Nobody can access us right now, so
Okay.
we're still hoping.
Okay, we'll open it up then to questions from the audience.
I think you can hear me. Right here in the front. In relation to behavior change campaigns related to this particular topic, what are the 2, 3, 4 areas that you would point to when we start looking at employing simple modifications for folks to start practicing better habits as it relates to ticks?
So you're talking specifically about prevention of tick-borne diseases. So there are a couple of strategies. One includes trying to avoid tick habitat, which is often easier said than done. And a lot of times the ticks are in residential areas where you simply can't avoid them. When that's not possible, we recommend using a repellent, specifically containing 20 to 30% DEET that you can apply to exposed skin or clothing, or using permethrin-treated clothing. Those are probably the simplest prevention steps you can take, but then it's also recommended that after coming indoors that you bathe and do a tick check. And any tick that you identify, you remove promptly to prevent the likelihood of the tick having the ability to transmit any of the pathogens that it's carrying. You can also tumble dry your clothing at high heat to try to kill any ticks that are still on your clothes. But most importantly, make sure that you do the tick checks, check yourself, your children, your outdoor gear, your pets, so that you're not bringing the ticks into your home for later exposure. Chris is going to add something else.
Yeah, I just wanted to follow up on that because it relates to the previous question in terms of tick removal. And as was mentioned previously, that's a very important process where you can reduce the risk of transmission. Not all the ticks that bite people are infected with a pathogen, but enough of them are so that getting that tick off you as quickly as possible reduces the risk of transmission. And different pathogens take different periods of time to transmit once the tick is attached. But the quicker you can get that tick off, the better you are. And the other thing is you need to be looking at your kids very, very carefully. And ticks will go to places that are hard to find. So you have to do a very thorough tick check when you've been in tick-infested areas. But that does minimize risk.
Okay. Dr. Schuckett?
One of you showed that nice origin of the Rocky Mountain spotted fever in Idaho, and in the maps, it really looks like, the Rockies don't have it anymore. I was wondering if you could clarify whether we're just seeing emergence of new vectors on the eastern, southern area, or if something changed in the western states to make Rocky Mountain spotted fever less frequent there.
Well, that's a great question, and I think what's really important to realize is that tick-borne diseases, like any zoonotic disease, they're dynamic processes. These things change. There's ebbs and flows in terms of the distribution, the prevalence, the frequency of infection. The disease was first discovered in the Bitterroot Valley of western Montana, but it existed in the eastern United States. It just wasn't diagnosed. It turned out that there was an epidemic in this small valley, and it was so dramatic that scientists from around the United States came to that valley to investigate, much the way scientists do now from CDC. So that's where it was discovered, and that's why it got the name Rocky Mountain spotted fever. It's really a misnomer because actually we now know that infection with Rickettsia occurs all throughout the Americas. We know that it exists in Brazil and Argentina and Colombia, as well as all the states in the U.S. And actually, most of the disease that we hear about now comes from the eastern U.S., where Dermacentor variabilis is the vector. What was very interesting is the emergence of this disease in eastern Arizona, associated with a novel tick vector. And that, I don't have enough time to go into the details of that, but that was a very intriguing process. And again, kind of the whole concept that these diseases change over time and new things emerge. We always have to be, prepared for new vectors, new pathogens, and new areas of emergence.
So over here, thanks all. Just to follow up on Chris's point, tough question, but what is your thoughts on the next emerging virus or bacteria from ticks? Can you predict anything? What are you expecting to happen?
Sure. I think there's a couple of ways to answer the question. if we say what virus is going to emerge and give us the largest number of cases in the shortest amount of time, something that we know about already, I think in my mind that's pretty clearly Powassan virus and its allies. And the reason for that is that it is in exactly the same transmission cycle that's driven the emergence of Lyme disease, human babesiosis, anaplasmosis, and so forth. So the question, I mean, there's a lot of interesting questions around that. Why have we not had a gigantic epidemic of Powassan virus encephalitis? And so, in a way, that's almost as interesting of a question as is that one really going to emerge? All of the kind of ecological factors that could drive that sort of an epidemic are there. Beyond that, are we likely to see more Bourbon virus, more Heartland virus? I don't know that I have a great answer for that, or the things that we don't know about yet, because we're not doing a particularly great job of going out and surveying ticks for viruses. Some of that work is being done, but it makes it hard to kind of predict what's going to come next.
So interesting, we've recently seen 2 fatal cases of Powassan virus, which is not common, but by any means, but that's a good prediction.
And Sharif, I think part of the answer to that question is the availability of assays to actually pick it up. And so if you don't look for it, you don't find it. And I think one thing that's very apparent is that we haven't discovered all the tick-borne diseases that are yet to be discovered. As Bobby mentioned, I don't know, more than half of them have been discovered in the last couple decades. So with the advancement of technological diagnostics that Bobby mentioned, I think we're going to find more. And then the other thing is you got to figure that there's not just 2 or 3 viral diseases, tick-borne viral diseases, or 2 or 3 rickettsial diseases. It's very likely that every one of these ticks has its own suite of pathogens, whether they're viral or rickettsial or protozoal. And we've found the most serious ones, those declare themselves usually pretty quickly, like Rocky Mountain spotted fever is very dramatic. So it's no surprise that was sort of the first one. It's the most lethal. But I think there are a lot of other, probably more subtle diseases out there that remain to be discovered.
Do we have any more questions from online? I don't want to completely neglect them.
No, we do, but let's go to the people in the room since we're not webcasting.
Okay.
Thanks for the great presentations. I noticed in some of your incidence maps there are some state-shaped holes of lower incidence surrounded by areas of higher incidence. So could you just describe surveillance systems for these types of diseases?
So most of the data, or almost all of the data that we have for these incidence maps are based on passive surveillance. And so you have all the limitations inherent in passive surveillance systems. So you may have some very diligent people reporting from a particular county in Florida or Oklahoma or Texas, and then people in other counties in other states that aren't necessarily looking or considering tick-borne diseases on their radar. So I think that's part of it. It's hard when you look at a map like that. It's hard to sort of say this county has it, this county doesn't, or this county has this high incidence, this county doesn't. I think it's better to look at it as a continuum and also just a general pattern. And that's why on the incidence map for spotted fever rickettsiosis, what I really wanted to emphasize was sort of the overall pattern, which was sort of a band that extended from Oklahoma to North Carolina. I didn't want to say, oh, look at this county in Tennessee. It's got huge incidence and the county next to it doesn't. The other thing is these are arthropod-borne diseases. Ticks don't fly. The infections can be very focal, where you find a cluster of cases in in a particular area, and then, 100 yards away, there aren't any infected ticks. So it's tricky. I hope that answers your question, Brian. Thanks.
Next question.
It sounds like prevention is a continuing and difficult challenge, trying to get folks to repeat something that needs to happen regularly. Given the success apparently that you had in the Southwest with the experience with dog tick collars, I'm struck, and I'm wondering if I'm going to get a couple of those dog tick collars for myself and use them to close up my trouser legs next time I'm hiking. Is wearable collar or bracelet or anklet strategies something worth exploring?
So I think the closest you would come is repellent or permethrin-treated clothing, but you hit on one of the toughest challenges.
I think I can get a dog tick collar pretty easily. I'm sure Amazon will ship them overnight.
It would be fashionable. The one thing to note, Chris had a notable success story with prevention of tick-borne diseases. Rickettsiosis sanguineus is a very different tick from the others that we've described. It's a single host, so it can be very effective to target domestic dogs. Whereas with your Ixodes scapularis-borne diseases, you tend to have the immature life stages feeding on small mammals, medium-sized mammals, the adults feeding on deer. So you have multiple hosts to keep track of, as well as those ticks are often found very close to people's homes and in areas where they like to recreate during the tick season. So repellent use, permethrin-treated clothing, and daily tick checks until the dog collar for people. Yep.
So on a number of the slides, especially having to do with the Ixodes range, the tick-borne disease range was actually much smaller than the Ixodes range. So what is the ecologic difference outside of the disease range where the tick still exists?
Yeah, that's a great question, and there are several differences. I think one of the key pieces that separates your risk in the north and the south is the behavior of the ticks. The southern Ixodes ticks are different enough from the northern Ixodes ticks that at one point they were two different species. And their host-seeking behavior is really different. In the north, the ticks more actively ascend the vegetation and more aggressively seek human hosts. Of course, humans are still incidental hosts, but you're more likely to encounter a tick in the north than in the south, strictly because of their host-seeking behavior. Infection rates are generally a little bit higher in the north than the south, particularly for Borrelia burgdorferi. The other pathogens are quite low in prevalence across the board. So, but largely differences in that host-seeking behavior.
Hi, thanks for a great presentation. You mentioned in one of the slides that doxycycline is the antibiotic of choice, but there are some challenges associated with it. It's not the first antibiotic a doctor will usually prescribe for somebody with a fever, for example. And it's also a challenge when it comes to pediatric populations. So I was just curious if you had any insights into doxycycline use in the United States, and if there was anything people should be doing if they know they've been exposed to ticks when they go to their doctor to maybe push them in that right direction.
Yeah, that's a great question, Jenny. So doctors are taught in medical school very strongly that you don't give tetracyclines to kids under the age of 8 because it stains the enamel and causes hypoplasia of the permanent teeth. Those studies were based on, that dogma is based on old studies, essentially women who were receiving tetracyclines in pregnancy for acne for multiple weeks. And what we know now is that a single short course of doxycycline in kids doesn't cause cosmetic staining of teeth. So The real push now, and what we've been trying to emphasize for the last 2 decades, is that doxycycline is the drug of choice. It's really the only drug that diminishes the fatality rates when administered in the first 5 to 6 days. So we've been pushing really hard to get physicians to be aware of that you give that drug regardless. It's not going to cause staining of the teeth if you give a 5 to 10 day course. I think that comes back to the very beginning in terms of how we prevent and control these diseases. And education and awareness are our most powerful tools right now. We are the CDC, but I think the control part of this is going to be very tough for years to come. Really, the key to all these tick-borne diseases is awareness and education of the clinicians who are seeing patients, as well as the public who are getting infected and presenting to their clinicians.
Thank you very much, and I'd like to take a moment to thank the speakers for excellent presentations. Thank you. And thank you very much for joining us, and we'll see you next month for CDC Public Health Grand Rounds.